During our experiment, we explored the effect of environment temperature on the growth rate and doubling time of Escherichia coli. According to the CDC, E. coli are a group of bacteria that primarily live in the intestines of animals as typical microbial flora. However, they can also be found in rivers and in other aquatic environments. According to Haig and Associates, E. coli does serve as a food source for some types of protozoa and bivalves. However, their presence often has a negative impact on human and animal populations seeking fresh, uncontaminated water. Pietrangelo addresses the presence of E. coli, saying that it can cause gastrointestinal distress, fever, kidney failure, and fatigue. Cashall and Associates discuss that climate change has begun to affect the ecology of the earth, including the temperature of rivers throughout the United States. This rise in temperature could cause an increase in the growth rate of E. coli as water temperatures begin to approach its optimum temperature, according to Albrecht. As such, we decided to conduct an experiment in which we introduced E. coli to various temperatures and then calculated the difference in growth rates and doubling time. This would theoretically model the alteration of E. coli population growth in the event that global climate change notably increased river temperatures. We hypothesized that water temperature has an effect on the growth rate and doubling time of E. coli.
We began by making tryptose phosphate broth, also known as TPB, to serve as the growth medium for the E. coli. Simultaneously, the lab strain of E. coli currently available in lab was restreaked on a TSA slant and incubated for 24 hours at 37 degrees Celsius. This process ensured that a fresh culture was used for each trial set. For inoculation, fresh E. coli culture was placed into 12 TPB tubes the morning of data collection. The initial transmittance of the tubes was then recorded using a spectrophotometer, which measures the amount of light that can pass through the tube. 3 of the tubes were then placed in each of the 4 designated locations that were held at a given temperature of either 4, 11, 22, or 37 degrees Celsius. The transmittance of the tubes was checked and recorded hourly for 12 hours in the initial trial, and then for 6 hours in the remaining 2 trials. Following data collection, growth rates and doubling times of the bacterial populations within the different temperatures were calculated. The graph shown here is the result of the log of absorbance at 600 nanometers plotted against the elapsed time for the average of the broth tubes at each of the temperatures. The values represent the clarity of the broth in the tubes, which represents the cell density in the tubes. This density increases as the population grows. The standard error bars represent the variation between the values. The overlap of the error bars on the growth curve can be attributed to the difference in cell amounts that were inoculated into each broth tube, as the size of the bacteria makes it so that the exact inoculation amount cannot be measured with the tools we had available. Taking the log of the absorbance produces the standard logistic shape of a growth curve. A typical growth curve will show 4 distinct phases: the lag, exponential, stationary, and death phases. These demonstrate the different rates of growth at the different stages of population growth in the culture. Because we only recorded data for 6 hours, the only stages seen in the graph are the lag phase and the exponential phase. The exponential phase is the period at which the growth rate greatly exceeds the death rate in the E. coli population. This is the portion of the graph that we use to calculate the average doubling time and growth rate. The graphs shown here represent the average doubling time and the average growth rate for the E. coli grown at each of the temperatures. Doubling time is the amount of time required for a population to double, while growth rate is the speed at which the population is growing. It can be noted that as the temperature decreases, the doubling time increases while the growth rate decreases.
According to UCMP, E. coli can survive at the average freshwater temperature range of approximately 4 to 22 degrees Celsius, but they thrive at temperatures around 37 degrees Celsius, according to Albrecht. Consequently, an incubator set to 37 degrees Celsius was used as the control. The other temperatures of 4, 11, and 22 degrees Celsius were chosen to represent a range of the river temperatures that they can survive. The exact temperatures were determined by the limited availability of temperature regulatory equipment. The tubes that were placed at these temperatures were placed in anaerobic conditions by tightly capping the lids. This therefore yielded growth rates that were lower than what would be expected for E. coli at aerobic conditions. However, the relationship of the growth rates at each of the temperatures is still indicative of the effect of temperature, since the conditions were consistent across all of the tubes. E. coli in river water all across the country tend to come from non-point pollution sources, which can include the manure of cows. The presence therefore acts as an indicator of fecal contamination as well as for general pollution, according to Botasova. As previously discussed, this presence can have harmful effects on the health of humans and animals. The cooler temperatures of rivers currently slows the growth of E. coli present, but if those temperatures rise due to climate change, the E. coli growth rate will likely increase as the doubling time decreases. Global warming is beginning to cause a rise in temperature in these rivers, as one article found that the annual mean water temperatures increased by 0.009 to 0.077 degrees Celsius in a year. With rising temperatures comes rising E. coli populations. In addition to the effects that rising E. coli would have on those ingesting the water, the ecology of the rivers would be impacted as well. Ultimately, our hypothesis that water temperature would have an effect on growth rates and doubling time of E. coli was supported.